Voice check.
Mic check.
Hello, everyone. Welcome to the 26th Journal Club at Antibuddies. Joining me today are two of our guests today. One is Cole Delia, and we also have Dr. Takeshi Zubata with us. So we'll start with Cole. A little bit about Cole. He runs a YouTube channel called Investigate, Explore, and Discover. It's all about science communication there, and I'll let Cole speak a minute about what's the channel about.
Thanks, Jatan. So my channel, Investigate, Explore, Discover, really focuses on providing summaries of scientific papers in an easy to understand format in about 10 to 15 minutes. So just a basic background that gives all the information you need to know about the paper and then a review of what that paper says.
Okay, thanks a lot, Cole. Guys, do check it out on YouTube, and it's also quite active on Twitter. And now we have Dr. Takeshi Subata, our presenter for the Journal Club. A little bit about Takeshi. He's a professor at Tokyo Medical and Dental University since 1996. He was the dean of School of Biomedical Sciences from 2003 to 2010. He received his PhD from Kyoto University, after which he worked at University of Cologne and Max Planck Institute for Immunobiology at Freiburg, Germany, as an Alexander von Humboldt Fellow. And then he worked at Department of Medical Chemistry at Kyoto University School of Medicine. He has been focused on learning about mechanisms for the regulation of immunity versus immunological tolerance of B cell lymphocytes to non protein antigens, especially roles of inhibitory B cell co-receptors in self tolerance that prevents development of autoimmune diseases such as systemic lupus erythematosus and Guillain-Barré syndrome. Takeshi, we're really glad to have you here today. And with that, I will pass over the mic to you so you can tell us more about your research.
Okay, cool. Thank you for the kind introduction. So good morning or the good evening or the good afternoon, everybody. So today I'm going to talk about our recent work on the Guillain-Barré syndrome. Okay, so can you see the slides?
Yes.
Okay, so today I'm going to talk about our recent work on the genetic studies on the Guillain-Barré syndrome. So first of all, I need to say that I'm an immunologist, but not the neurologist, and also I'm a MD, but I never saw patients with GBS. So probably you are interested in why we worked on GBS, though I never saw patients with GBS. So today first, I'm talking about our work first, and then after that, I will explain you why we worked on GBS. So the Guillain-Barré syndrome, or GBS, is an acute neuropathy that involves autoimmune mechanisms. So the neuropathy is a disease where peripheral nerves are damaged. That causes motor and sensory paralysis. If the respiratory muscles are involved, so then the patients need mechanical ventilation for survival. And in GBS, paralysis progresses over hours and days. So that's why it is acute neuropathy. And this is what is understood about the pathophysiology of the GBS. So quite often there's preceding infection by the microbes such as Campylobacter jejuni or the Haemophilus influenza. So those bacteria express glycolipids on the cell surface, which have structural similarity to the gangliosides expressed on the human neuronal cells, such as GM1 or the GQ1B. And there's the antibody response to the microbes, including the production of the anti-glycolipid antibodies. Some of the some of the antibodies to the glycolipids expressed on those microbes cross-react to the gangliosides because of the structural similarity. And then the anti-ganglioside antibodies bind to the gangliosides expressed on the neuronal cells. Then this causes the damage of the neuronal cells and causes the neuropathy. So yes,
Yes,
JT?
I have a quick question.
Yeah.
We hear about, whenever I think about an autoimmune neurological disorder, the first one comes to my mind is multiple sclerosis. So compared to multiple sclerosis, what would be that one distinguishing factor that differentiates this disease?
Yeah, so the GBS is disease whose prognosis is very good. So of course, patients get sick, but without any treatment, patient recover. But in the case of the multiple sclerosis, it's the progressive disease. And second thing is the anti-ganglioside antibody is a major cause of the disease in GBS. But in multiple sclerosis, what I understood is that the T cells are the major players.
Okay, thanks a lot.
Okay. Okay, so then what is gangliosides? So this is the sphingosine synthesized in basically all the cells. And when the fatty acid is conjugated to this nitrogen, so then this is ceramide. And the various groups are conjugated to this oxygen in the ceramide, and these are called the head groups. And the sphingomyelin contains the phosphocholine as a head group. And if head groups are the sugars, then they are called glycolipids. The galactosyl ceramide contains the galactose as a head group. Sulfatide contains sulfogalactose as a head group. And the glucosyl ceramide contains the glucose as a head group. And if the head group is contains the multiple sugars, including sialic acid, then the compound is called gangliosides. So gangliosides contains the sugars, and there must be at least one sialic acid. So this is the phosphatidylcholine, which is major component of the plasma membrane. And you can see the structural similarity between the phosphatidylcholine and the sphingomyelin. So this is why the sphingomyelin is located also in the plasma membrane, and that is also the case for the other glycolipids. So this is the plasma membrane, and the gangliosides are located on the outer leaflets of the plasma membrane. Therefore, the sugar chains are exposed to the outside, and the gangliosides are enriched in the plasma membrane microdomain, which is called the lipid rafts. Okay, so the gangliosides are the groups of the chemical compounds that are heterogeneous in the sugar chains. So there are many ganglioside species, and the expression of the ganglioside species depends on the cell types. So this is result thin layer chromatography of the glycolipids from the various different cell types. So this is the oligodendrocyte. This is the fibroblast, and this is the cerebellar granule cells that are the neuronal neurons. And you can see that the expression of the glycolipids species are different among the different cell types, and only the neuronal cells expressed those molecules that are the glycolipids containing the larger numbers of the sugars. And the antiganglioside antibodies in GBS recognize those gangliosides that are specifically expressed in neuronal cells. So therefore, the symptoms is specific to the neuronal disturbances. Okay, so we did the genetic analysis. So why people do the genetic analysis? Of course, you know, you want to identify the genes that are involved in the diseases, but quite often in polygenic diseases, the contribution of each the genes associated with a disease is have a very small effect. But nonetheless, the identification of the disease-associated genes can elucidate the pathogenic mechanisms in the various diseases. So this is the list of the genes associated with systemic lupus erythematosus, and you can find the nucleases like the TREX1, DNase1, DNase1L3, and also the receptors for the nucleic acids, nucleic acid sensors, such as the TOL7 and the IFIH1, and the presence of the genes involved in the DNA nucleic acid metabolisms, and also the receptors for the nucleic acids are quite specific to the systemic lupus erythematosus, and this suggests that the involvement of the response, hyper response to the nucleic acid as critical cause of the pathogenic cause of the systemic lupus erythematosus. So then what about the genetic variants associated with the GBS? So in families with the GBS patients, the incidence of GBS in other members of the family is significantly higher than non-family members. So this clearly indicates that the genetic factors are involved in the development of GBS. However, there are many studies on the association of GBS with major histocompatibility complex (MHC), but so far there was no association shown of the GBS with MSC. So eventually, this is an exception as an autoimmune disease because the autoimmune diseases involve the immune responses. So then almost all the autoimmune diseases associated with MSC. Only the exception is GBS. So there's no association of GBS with MSC. And also, there were many candidate gene approaches to identify the non-MSC genes associated with GBS. So there are papers describing the association of the CD1 with C-gamma receptor, others with GBS, but the results are controversial. And there were multiple papers describing the association of TNF alpha with the GBS, but of course, TNF alpha is a crucial player in the inflammation. This doesn't tell us any specific pathogenic mechanisms of GBS. And there was the genome-wide association study, GWAS, but no significant association was found. So in GBS, it is clear that there must be the genetic factors, but so far no obvious genetic factors was discovered. So probably you know the difference between the common disease variants and the rare disease variants. The common disease variants are the variants where minor allele frequency is more than 5%. In contrast, the rare disease variants, the variants where the minor allele frequency is much less, so normally less than 1%. And often the rare disease variants often the rare disease variants are population specific. So because of the frequencies, the common disease variants can be identified by GWAS, but the rare disease variants can't be identified as GWAS because the frequency is very low. So you need to do the DNA sequencing to identify the rare disease variants. So the odds ratio is quite low for the common disease variants. So it's normally less than 1.5. Means that the risk for the disease is increased by at most 50% if you have the particular variant. But the odds ratio is much higher in the rare disease variants. And also the penetrance is different. So the common disease variants show the low penetrance. So then the prophylactic intervention is unlikely. In contrast, the penetrance of the rare disease variants is often high. And so then you can do the prophylactic intervention using the rare disease variants. And also the functional relevance is unlikely in common disease variants, but the relevant functional relevant in the case of the rare disease variants. And then the contribution to the disease etiology is unlikely in common disease variants, but the contribution, there's the contribution to the disease etiology if you have the rare disease variants. And the common disease variants may suggest the candidates for rare variant search. And the rare disease variants have effect. The effect of the rare disease variants may be modified by common variants. So the polymorphisms identified at GBAS explain only a fraction of the genetic factors in many of the diseases. So this suggests that most of the contribution by the genetic factors are made by the rare disease variants, and if you can identify the rare disease variants, then you can address the etiology of the diseases. So then we did a search for rare variants associated with GBS. So now I'd like to introduce you about the inhibitory B-cell co-receptors. So the B lymphocytes express various inhibitory receptors that inhibits the antigen receptor signaling. So when BCL interacts with the antigens, then the BCL associated kinases, such as LAN or the SIG, get activated. And those kinases phosphorylate downstream signaling molecules, thereby inducing the BCL signaling. But the LAN phosphorylates inhibitory receptors as well. So in the cytoplasmic region of the inhibitory receptors contains the items, so inhibitory tyrosine motif, and once the tyrosine in the items are phosphorylated, then the phosphorylated items recruit and activate phosphatases such as the SHP1, SHP2, and the SHIP1, and those phosphatases they dephosphorylate the activated signaling molecules, thereby inhibit the B-cell receptor signaling. The CD72, so one of the inhibitory B-cell receptors, associates with the SLE in both human and mice, and the CD72 inhibits the B-cell, inhibits the development of SLE, because the CD72 deficient mice develop lupus-like disease spontaneously. And some time ago, we showed that the CD72 recognizes lupus self-antigen SMRP and inhibits B-cell response to SMRP. So CD72 appears to recognize lupus self-antigens and inhibits the B-cell response to the self-antigen, and inhibits the development of lupus. So the inhibitory B-cell co-receptors may be important in the self-tolerance. And there are two inhibitory co-receptors that recognize sialic acid. So as I mentioned, the gangliosides contain sialic acids, and the CD22 or SIGLEC2 and the SIGLEC10 in mice, which is called in mice, this is called the SIGLEC-G. Both these inhibitory co-receptors recognize sialic acid. So then we thought that maybe those receptors recognize gangliosides and regulates the B-cell response to the gangliosides. So then we did the analysis of the genes for these inhibitory co-receptors. We did this in collaboration with Professor Kusunoki at the Kindai University Hospital. So Professor Kusunoki is the expert of GBS in Japan, and we recruited the 31 patients with GBS and 6 patients with the MFS. MFS is what was? This is the variant. I mean the subtype of GBS. So yeah, Miller-Fisher syndrome. And we also have those patients as the control patients. So the SIGLEC10 is quite polymorphic. So there are many, quite a large numbers of the polymorphisms in the SIGLEC10 genes. But most of them are not associated with GBS. So this is the list of the polymorphisms of the SIGLEC10 that are not associated with GBS. But we found two polymorphisms in the exon 2, which encodes ligand binding domain associated with GBS. One is the variations that confers the replacement of the arginine 47, arginine at the position 47 by glutamine. The other is the variations that causes the replacement of the alanine at the 108 by valine. And in the GBS patient, the more than 10% carry these variations, whereas none of the control patients have these variations. And there's statistical significance. And the R47Q and the A108V have the linkage disequilibrium. So this means that all the patients who have the R47Q also have the A108A, 108V. So we couldn't find any patient who has either R47Q or the A108V. All the patients who have R47Q have A108V. Okay. So this is the data from the, okay, so yeah, Cole, do you have a question?
Hi there. Yeah, I have a quick question. In your last slide, you included the MFS patients with the GBS, and there were 5 of them, I believe, from the patient cohort. So are those mutations that you see all from the MFS population, or were they interspersed throughout people that had GBS as well?
No. Most of them are GBS because we have more GBS patients than the MFS. But we had the one MFS patient who has this mutation. Okay.
Okay. Thank you.
Okay. So this is the data from the Thousands of Genomes project, and we just looked at the frequencies of this mutation in the various ethnic groups. So if you look at the whole population, so then R47 frequency of the R47Q is very low, less than 1%. And none of the Africans and the Native Americans, Europeans, and the Southern Asians has this mutation. This mutation is found only in the East Asians. And if you look at the subpopulations, 1 to 3% of the alleles in the population are the R47Q mutation in the Chinese dye. So this is ethnic group living in the southern China, and also southern Han Chinese, Japanese, and the Vietnamese. And there's no Han Chinese who has this mutation. So therefore, this mutation is located only in South China and Japan. And the same is true for the A108V. So this is the origin of the Japanese. The Homo sapiens came to the Japanese archipelago through the 3 routes. One is the northern route, from Siberia, Sakhalin, and Japan. And this is the Korean peninsula. And some of the people came from the Korean peninsula. And the third route is the southern route. So the people came from Taiwan and also the Ryukyu Islands and then Japan. So because the R47Q and the A108V are found only in South China and Japan, so probably the people came from the southern route may brought this mutation to Japan.
That's such a nice speculation. Hey.
Thank you. Okay. So then we addressed whether these mutations modulates the function of the SIGLEC10. So to address this question, we made the recombinant protein of the SIGLEC10 that contains the extracellular region of SIGLEC10 and the FC region of human IgG1. And also we generated the wild type molecule and also the recombinant proteins that contain the R47, either R47Q, A108V, or both mutations. And first we looked at the binding of these the recombinant proteins to the cellular ligands expressed on the human cells. And the mouse CD22FC is the control because the mouse CD22 recognizes only the salicylic acid species that are expressed in mice. So then the mouse CD22FC doesn't bind to human cells. So this is the negative control. And you can see that the both the wild type SIGLEC10FC and the SIGLEC10FC with the A108V bind to the MCF7, whereas the R47Q or the double mutant fail to bind to MCF7 cells. And these bindings are abolished if you treat the cells with the neuramidase. Neuramidase is an enzyme that they degrade the salicylic acids. So this result clearly shows that the R47Q, but not the A108V, impairs binding of SIGLEC10 to salicylic acid containing cellular ligands. So then we next looked at the binding of these recombinant proteins to gangliosides. So we did this analysis by ELISA, and you can see that the wild type SIGLEC10FC can bind to various gangliosides. But if you look at the mutants, mutants with the R47Q fail to bind to these various gangliosides, and that is also the case for the double mutant. But the mutant with the A108V binds to the various gangliosides as well as the wild type. So again, R47Q but not A108V impairs binding of SIGLEC10 to gangliosides. So these results clearly indicate that the SIGLEC10 R47Q is a loss of function mutation. So, but the question was how R47Q inhibits the binding of SIGLEC10 to the gangliosides, because the arginine 47 is distant from the ligand binding site of SIGLEC10. So this is the structure or molecular structure of the SIGLEC10 ligand binding domain. And this is the arginine, which plays a central role in the ligand binding. So the salicylic acid is bound at this site, but the arginine 47 is a bit apart from the ligand binding domain. So then that was a question. why arginine 47 mutation at the arginine 47 impairs the ligand binding. So the crystal structure of the SIGLEC10 has not yet been solved, but there are SIGLEC5 and SIGLEC8. The structures were solved. So then we looked at the structure of those molecules, structure of the ligand binding domain of the SIGLEC5 and SIGLEC8, and found that they were structurally quite different. So in SIGLEC5, so SIGLEC5 is shown in cyan, and the SIGLEC5, in SIGLEC5, the tryptophan 52, the stuck to arginine 127. The amino acid at the position 37, which corresponds to the tryptophan 52 in the SIGLEC8, is glutamine, and the glutamine can't form the pi-pi interaction with the arginine. So then the Q37 can't stuck to arginine 112. Instead, tryptophan 40 at the position of 40 stuck to arginine 112, and the tryptophan is corresponding to the tryptophan 55. So as you can see here, the tryptophan 55 doesn't stuck to the arginine. Instead, the tryptophan 52 stuck to the arginine 127 here in SIGLEC5. But in the case of SIGLEC8, the tryptophan 40 corresponding to this tryptophan stuck to arginine 112. So then you have quite a big difference in this region. So then what about the SIGLEC10? So the arginine 47 is eventually corresponding to the tryptophan 52 in SIGLEC5. So then probably the arginine 47 may interact with the arginine 122 and forming the SIGLEC5-like structure. But if the arginine 47 is replaced by glutamine, so then the SIGLEC10 can no longer have the pi-pi interaction between the glutamine 47 and the arginine 122. Instead, the tryptophan 50 may form the pi-pi interaction with the arginine 122. So this may change the structure from the SIGLEC5-like to SIGLEC8-like by the arginine 47Q mutation. So is this clear?
Yeah.
Okay. Okay. So do you have a question?
Yes. So far, the data you have shown, that alanine to valine mutation, is it just coincidental and it has no functional relevance?
Yes, because the SIGLEC10 recombinant protein with the A108V alone can bind to the gangliosides as well as the wild type. So we are thinking that the A108V is just a coincidence because of the linkage disequilibrium. So they are located too close. So then they can't be separated. Okay. So then this is the modeling of the SIGLEC10 ligand bind domain in SIGLEC5-like structure and the SIGLEC8-like structure. So as I mentioned, the wild type SIGLEC10 may form the SIGLEC5-like structure, whereas the SIGLEC10 R47Q may form the SIGLEC8-like structure. So So then the tryptophan 50 is located here in the wild type, but this stucks to the arginine 122 in the R47Q mutant. And this shift causes the structural alteration of the flanking region. And if you little bit rotate, then you can see that the flanking region of the 47Q shows the structural alteration. So this is the structure of the SIGLEC5-like, and this is a structure of the SIGLEC8-like. So if you have the SIGLEC8-like structure, so then this region protrudes towards the ligand binding domain, and then disturbs the ligand binding of the SIGLEC10 ligand binding domain. So therefore, the R40Q mutations the induces structural change of the flanking region, thereby inhibiting the ligand binding of SIGLEC10. So to summarize, SIGLEC10 rare variants, arginine 47Q and the A18V are accumulated in GBS. And these variants are population specific. So Japanese and Southern Chinese have the mutations, but no other people have these mutations. And the R47Q impairs binding of SIGLEC10 to ligands, including gangliosides. And the arginine 47Q alters the ligand binding site by globally altering the structure of the ligand binding domain of SIGLEC10. And the SIGLEC10 may inhibit production of the anti-ganglioside antibody by recognizing the gangliosides. And the identification of SIGLEC10 R47Q may be utilized for prophylaxis because if you have, for example, the raw chicken, then you have a risk to get the infection with the Campylobacter. So then if you know that you have the SIGLEC10 R47Q, so then you should avoid eating the raw chicken, for example. Okay. So now I want to talk about why we are interested in GBS. So because the immune responses to the non-protein antigens is distinct from the immune response to the protein antigens. So as all of you know, the T lymphocytes recognize the peptide the loaded on the MHC molecules. So therefore, T cells recognize proteins. But the T cells can't recognize other macromolecules, such as the nucleic acids, polysaccharides, and the some of the glycolipids. So the glycolipids are basically presented by the some specific MSC molecules or the MSC-like CD1 molecule. And some of the glycolipids can be recognized by T cells, but the T cells can recognize only glycolipids that contains only small number of sugars, like the monosaccharide. But the gangliosides have the larger numbers of the sugars, so then the T cells can't recognize gangliosides. But the B cells can recognize basically all the macromolecules, including nucleic acid, glycolipids, polysaccharides. So this can be easily understood because you can find the antibodies to nucleic acids, antibodies to glycolipids, antibodies to polysaccharides. So the B cells can recognize all those macromolecules, but the T cells can recognize only proteins and some other molecules, but not all. So basically, the activation of the immune responses, so B cell responses to protein antigens depends on T cells. So T cell help is required. And also the self-tolerance to the protein antigens largely depends on the T cells. So largely depends on T cell tolerance. This means that the B cell response to non-protein antigens requires mechanisms distinct from the B cell response to the protein antigens. And also B cell tolerance to protein antigens requires mechanisms distinct from the B cell tolerance to protein antigens. So we are interested in the molecular mechanisms for the B cell response to the non-protein antigens because they must be quite different from the immune responses to the protein antigens. So what about the immune responses and tolerance to the nucleic acid related antigens? So this is the example of the nucleic acid related antigens. So SMRP is one of the lupus autoantigen. So the anti-SMRP antibody is a disease-specific antibody, autoantibody. And if you have the B cells reactive to the SMRP, so then the recognition of the antigen induces the B cell receptor signaling, and also the antigen is internalized after the BCR after the interaction of the BCR, and then the antigen is transported to the endosome, and there's the nucleic acid sensors, so receptors for the nucleic acids in the endosome, such as the Toll7 that recognize the RNA, and then the RNA component in this self-antigen interacts with Toll7 and induces the activation signaling. So then the autoantibody is produced by the B cell receptor signaling and also the TLR7 signal. This is the structure of the SMRP. So it's a complex of the RNA and also the proteins. So the nucleic acid sensors are the mechanisms of the B cell, specific mechanisms for the B cell response to the nucleic acid antigens. What about the tolerance? Some time ago, as I mentioned to you, some time ago we showed that the inhibitory co-receptor, inhibitory B cell co-receptor CD72 recognizes SMRP and inhibits B cell response to SMRP. So the CD72 is so the CD72 is CD72 contribute to the B cell tolerance to this self antigen. Okay. So then, so CD72 recognizes the SMRP and inhibits B cell response. So as I mentioned, the gangliosides contain sialic acids, and the sialic acids can be the self-motif that inhibits immune responses. So the sialic acids is quite special as a sugar. So it's the 9-carbon acidic sugar and located at the end of N-glycan, O-glycan, and glycolipids. So basically, if you look at the cells, the most outer parts are occupied by sialic acids. So can be recognized by the various other cells, and the structurally diverse. And the sialic acids are present in vertebrates, but not in plants, fungi, algae, and bacteria. And the sialic acids inhibits immune responses by complement inhibitor factor H. So it activates the complement inhibitor factor H, and also by SIGLEC. So as I already introduced to you, SIGLEC are the membrane molecules that recognize sialic acids and expressed by various immune cells. And the B cells express SIGLEC10 and the CD22, and both of them are inhibitory receptors. So SIGLEC also recognize cytokine acid and then inhibits the activation of the immune cells. Here I wrote the exception. So some of the bacteria express cytokine acids, but in this case, cytokine acid is the virulence factor. Example is the group G Streptococcus, and people think that some of the pathogens they acquire the ability to produce cytokine acids to evade from the host immune responses. However, there was no evidence supporting the role of the SIGLEC in self-tolerance. Yeah, so this is the list of the SIGLECs. So there were around 10 members, both in human and mice, and there was significant difference between human and mice. Many of them are the inhibitory receptors, and each SIGLEC are expressed in the different cell types in the immune cells. And among them, the CD22 and the SIGLEC10 are expressed on B cells. Okay, so then this is why we did the genetic analysis of the SIGLECs in GBS, because first of all, gangliosides contain cytokine acids, and the gangliosides are not recognized by T cells, and there's a distinct mechanisms for the antibody production to gangliosides, because the B cell responses, immune responses to gangliosides are not restricted by MHC, and then there must be distinct tolerance mechanisms to the gangliosides, and that may be. So we thought that may be related to the cytokine acids, and we thought that we can show that the involvement of the cytokine acids in self-tolerance by analyzing the production of the anti-ganglioside antibodies in GBS patients. So this is the summary slide. So the self-tolerance to the protein antigens largely depends on the T cell tolerance, and the B cell responses and tolerance to the nucleic acid containing antigens and the glycolipids are distinct from the protein antigens, and for the activation of the B cells reactive to the nucleic acids, the nucleic acid sensors like the TOL7, TOL9 are involved, and for the tolerance, the CD72, which is one of the B cell inhibitory co-receptor, recognize the self-antigens and inhibits the B cell responses. And for glycolipid self-antigens, here we show that the SIGLEC10 binds to gangliosides, and probably the SIGLEC10 is involved in the self-tolerance to the ganglioside specific B cell responses. So although we do not know what is the activation mechanisms of the B cells to the glycolipids, so this is the future project. So this is my group. The work on the GBS is done by Amin Alboljan, and also the former members Matthew Rutulich, Soha Goma, and the Xuexin Li. And this work is done in collaboration with the Professor Kusunoki, who is the expert of the GBS in Japan, and also the other people listed here. So thank you very much for your attention.
Thanks a lot, Takeshi. I'm working with TLR7 signaling right now, and I think this was unexpectedly very, very useful journal club for me. I came here to get just general knowledge, and right now I'm going back with very specific knowledge. Like I had never heard about CD72, which I feel like I should have known about CD72 before. For a fact
Yeah.
that, do you know if it's expressed in non B cells?
Yeah. So the CD72 is most highly expressed in B cells, but the CD72 is also expressed by the innate immune cells like the macrophages and NK cells and DCs, but the expression level is much lower than the B cells.
Okay. So if I could summarize the concept that I'm taking back, it's that a B cell normally will go and maybe it will see the ganglioside on a neuron, but then as soon as it sees the ganglioside at the same time, other co-receptor like SIGLEC10 would also see the ganglioside and it will stop the activation.
Yeah.
And that's
Yes.
what's
Yes.
happening here.
Yeah.
Okay.
Yes.
It's a very nice concept. Yeah.
Thank you for your presentation. I just have some clarifying questions. So when people that you found with that arginine to glutamine mutation, was their prognosis worse or more severe upon identification of GBS?
Yeah. So it's a very important question. So we have the limited numbers of patients, but there are roughly two distinct types of the GBS. One is caused by the damage to the neurons, and the other is caused by the demyelination. And yeah, so yeah, so the AMAN, so acute motor axonal neuropathy, is caused by the direct damage by the antibodies, and the AIDP is a subtype of the GBS in which the demyelination plays a role. And we looked at the subtypes of the GBS, and then basically we found that there's no AIDP in the patients with the R47Q mutations. Although around half of the GBS patients in Japan are the AIDP, and as you can see here, the anti-ganglioside antibodies are more associated with AMAN than the AIDP. So what this tells us is that among the GBS, so certainly the GBS is diverse, and some patients more dependent on the anti-ganglioside antibodies and then develop the AMAN, and those patients with the R47Q tends to develop the AMAN, which is more associated with anti-ganglioside antibody.
Okay. And just a general thing really quick. So when being diagnosed with GBS, these people are getting infected most likely with Campylobacter jejuni, which then activates B cells in a similar fashion. So that way they then recognize the neurons or the myelin through some sort of molecular mimicry. And that's what causes the antibodies to then attack the myelin sheath.
Yeah, yes. Yeah, so eventually the myelin sheath doesn't express gangliosides. So gangliosides are expressed by neurons.
Okay. Thank you.
Mm-hmm.
So it is molecular mimicry with the infections.
Yeah. Yes.
Okay.
Yeah. So this is one of the typical example of the molecular mimicry.
Okay.
So Maria.
Yeah. Yeah, I have a question because I've recently read some articles about using some I don't know sugar substrates or something like that in modifying like glycosylation processes in treating some autoimmune diseases. And I was wondering if increasing the sialylation in those gangliosides would somehow help binding to SIGLEC10 or not.
Yeah, so we do not have any data. Okay, so it's very difficult to address the role of the sialic acids in these responses. So I have to say that our data is quite indirect. Okay, so I think it must be the further works must be done by, for example, establishing the mouse or the in vitro systems where the SIGLEC10 function can be evaluated more closely. Yeah. So
Yeah.
to your questions, the quick answer is that I do not know.
Thank you.
Okay, are there any other questions? If not, okay. So I will end the statement saying that we recently did an Immunology 101 podcast episode. Dara was there, and we talked about how B cells have little role in tolerance, and it's mostly T cells. And now I wish I could go back and add this thing that no, there are glycolipid and lipid mediated tolerance that B cells mediate.
So it depends. Depends
Yeah.
on the types of antigens.
Right. So I feel like that podcast is already dated. We'll make an update very soon. But thanks a lot, Takeshi. This was a wonderful, wonderful learning experience for all of us. And yeah, good luck with your research. And
Okay.
to everybody listening to us, we'll be back in 2 weeks with another journal club, possibly the last journal club of this year before we sign off for 2022 again. Meanwhile, thanks you all for joining. This video will be available on YouTube and Facebook for viewing later. See you all. Bye bye.
Thank you. Bye bye.